Thank you very much for inviting me here again. I was here last year and I enjoyed it greatly. I also want to state my condolence to Henrik Konakowski, who was a sales agent for ChemDiff. This young boy just died a week ago on cancer, and that gives us a reminder that we're not only doing research for scientific curiosity and not only research for industrial interests, but in the end we do research to save lives. And some of the work that we're doing luckily relates to that and hopefully will lead to new forms of treatment that could contribute at least to save life. Let me see whether that works. So I just want to start with a more general part about druggability. I want to remind you that only 2% of human proteins interact with current approved drugs. So there's a huge part of the proteome that is not addressed by drugs. 10 to 15% of human proteins are disease-causing, and about 10 to 15% human proteins are supposed to be druggable. Now that could cause a lot of excitement that there's still a lot out there to be discovered, but unfortunately these things are not overlapping, and we do not really know how much exciting drug targets are around that need to be explored. We certainly know that there are some protein families that are overrepresented in drug discovery, like kinases, phosphatases, GPCRs, but still there are ongoing interesting observations and discoveries. But there are also some protein families that are underrepresented in drug discovery, like ARTs, ubiquitin ligases, ubiquitin proteases, catalytic enzymes for rare post-translational modifications, etc. So I think at current nobody really knows how much blockbusters are around there to be discovered, but certainly there is still substantial room for both academic and industrial discoveries. And that brings me to the interaction between academics and industry. I include this slide and the following slides because I noticed that there is a strong interest here in establishing industrial activity around novel therapeutics. And there's obviously a question mark how good the match is between academics and pharma. Academics are very excellent in being creative, innovative, trying new things and discovering entirely new tools and mechanisms, while pharma is increasingly closing up their early stage discovery pipeline and rather focus on late stage development and buy-in of drugs that already have reached clinical trials. So there may be a match, and if we look at the enormous costs that are involved in bringing a drug or a hit from the discovery stage to an FDA-approved compound, is both enormously high in costs, around $1 billion, and also takes a very, very long time, on average 16 years. So it is rather unfeasible that a purely academic effort can reach a drug through all these steps. However, both academia and industry very strongly realize that there is high value in the work that academic laboratories can deliver, and industry is looking very intensely out in the academic world for research groups that are innovative, that develop novel assays, that are good at hit identification, especially in collaboration with such excellent screening units that, as the one that we just heard from my very good colleague Jens von Gries. Industry is very keen in interacting with academia on novel disease models, biotarget validation, and to some but lesser degree in hit-to-lead efforts. So how does that then all pan out in terms of targets? It is quite interesting. This is a recent review to see that academia here, you see that compared the US academic efforts and UK academic efforts as to pharmaceutical efforts in target identification, both are roughly matching. So academia is doing, by that measurements, extremely well compared to the much richer industry in identifying interesting targets. Top hits are, or top disease areas are cancer, cardiovascular disease, but also metabolic disease, diabetes, endocrine disease are very prominent, as are infectious disease. This part I find rather interesting because I hear a lot about IP and securing IP and public-private partnerships between academia and industry. Now, if you look at the funding that academia uses for their discovery, you will see that very little of that funding really comes from IP or public-private partnerships. And much of the funding comes from classical public funding like research councils and non-profit organizations. So that was the general comment on academia as really getting very important players in drug discovery, but also some grain of salt on the interactions between academia and industry, which doesn't mean that the partnership isn't absolutely necessary to carry through drugs in clinical trials. The second part of my talk, I want to talk about the signaling system that I've been spending all my scientific career upon, and that is basically two signaling systems, Wnt and hedgehog signaling. Both signaling systems were discovered by pure academic curiosity. Nobody was ever thinking that this could have any commercial potential, and it was purely curiosity driven. Here I show you two in situ hybridizations that show the first expression patterns of these genes in zebrafish, and I also show you that these signaling cascades, as they are extremely powerful, and if they're messed up, for instance in the case of hedgehog signaling, if you have a deficiency in hedgehog signaling, you will get a fusion of the midline and one-eyed embryos. And if you get an overexpression of Wnt signaling, you get an axis duplication that can lead to animals that have two heads or even three heads and two body axes. Now if we move 20 years in time since these early discoveries were made, those pathways have become very prominent in drug discovery. In hedgehog signaling, there is now one approved drug, vismodegib, that has reached patient care for basal cell carcinoma. And there is also a number of natural products that can inhibit the pathway, such as cyclopamine, that is found in a certain form of lilies. Now, Wnt signaling is very interesting because there is no approved drug so far, and that's rather remarkable because it is a very powerful signaling pathway. But again, there are a number of natural products like flavones, for instance, that are found in green tea that have a very fundamental impact on Wnt signaling as such. Now, why is Wnt signaling so important? It's important in a number of areas. I want to highlight, first of all, cancer, and aberrant Wnt signaling has been found in a large number of different cancer types, including colon cancer, prostate cancer, non-small cell lung cancer, breast cancer. And Wnt signaling is considered to be as one of the few central signaling cascades that are dominant in cancer development, often as an initial step, as we see in colon cancer or colon adenoma formation. But Wnt signaling has multiple forms, multiple functions in cancer. It's involved in tumor initiation, often by mutations in a structural protein that is called APC. It's directing tumor growth, it's directing migration of tumor cells, and in the last years we start to get aware that it's also involved in directing cancer metabolism that is called Warburg metabolism, and possibly also in immune response. So it's a whole panel of different features that are somehow and often in an interactive way influenced by Wnt signaling. No wonder that it is an important pathway for drug discovery. But Wnt signaling has not only something to do in cancer development, it's also involved at very early differentiation steps and stem cell homeostasis. For instance, if you have an ES cell in the undifferentiated state, you want to have a median level of Wnt beta-catenin signaling. If the signaling drops, you get neuroectoderm. If the signal increases, you get endoderm. In both cases, you get differentiation. Now, Wnt signaling is also involved then in differentiation, as I already told, and this can be is used in nature, of course, but can also be used in artificial differentiation system where you need combinations of different signals, including Wnt and hedgehog signaling, either being activated or blocked to reach a particular cellular phenotype. In this case of the scheme, it is islet cells of the pancreas that, as we all know, direct energy metabolism and insulin production, in particular beta cells. But this Wnt signaling is also then involved in tissue regeneration, in aging, and it has lately been implied in metabolic disease. A lot of reasons to develop drugs that can address this pathway. I want briefly to explain you the pathway. The pathway is in principle very simple. There is a protein that is called beta-catenin that is at the junctions of cells and gets released from there and migrates into the nucleus. And if it does so, then the signal is active. But if and this is the case when the Wnt signal comes into the signalosome. When the Wnt signal does not come into the signalosome, then a destruction complex or degradosome is formed, and that effectively keeps beta-catenin from migrating into the nucleus, and instead beta-catenin gets phosphorylated, recognized by E3 ubiquitin ligase, and led to the proteasome to be destroyed. So that's in very simple ways how the signaling cascade works. Obviously, things are never that simple in nature, and here I show you more advanced scheme how Wnt signaling works. And in particular, I want to point out that neither the signalosome is as simple as I have shown in the first slide, and there are multiple ways of activating the signalosome. And lately, a second morphogen has been found, R-spondin, that is very crucial in the activity of the signalosome. But also the destruction complex or the degradosome is by no means as simple as one initially thought, and it turns out that it is a mesh of proteins that interact with different kinetics. And so far, although we have started to get the structure of certain domains of this network of protein domains, we have by no means any idea how the degradosome in its complexity looks like, and also we don't know too much about the kinetics between the different members of the degradosome that all will have influence on how Wnt signaling or beta-catenin reach the nucleus and how active Wnt signaling can be. Now, this is the basis for our drug discovery program. We approached Jens some years ago whether he was interested to initiate a drug discovery project with us. He was, as always, very enthusiastic and has been one of our best colleagues, if not our best colleagues of all, in helping us to set up assays and run a high content screen, which led to a number of hits. Here I show you one. That is JW55. I don't want to go into any other details than saying that we get very extensive information on all the positive hits that allow us very carefully to select out false positives and move on to the very important step in academic drug discovery, and that is target identification. All our efforts make very little sense if we're not working with unique targets that haven't been addressed by the pharmacological industry, and unique targets that still lack drugs and understanding how drugs on these targets work. If you find a unique target, you're into an interesting ball game. So what we did was to identify the target of of our lead hit, and we were very enthusiastic to find out that it is catalytic enzyme of a family that is called the ART family or PARP family. That is a family of proteins that does post-translational modifications, and they're called poly-ADP ribosylase family. And the main member of this family, PARP1, is a nucleoprotein that has been very heavily addressed by the pharmacological industry, and that just has been registered one drug for clinical use last year, while the protein that was pulled out in the affinity column with the drug that we identified in Berlin turned out to be a different member without known drugs yet in any clinical trials or use, which is called tankerase. Now, tankerase is an interesting protein in this family because it's both a structural protein that through its anchoring domains can form active protein-protein networks and interactions. But it's also a catalytic protein that can use energy or redox metabolite to form a post-translational modification in target proteins. We tested the protein in an animal model. I already explained you the axis duplication model. In this case, we used Xenopus to see whether the drug really specifically could revert an induced axis duplication by overexpression of Wnt signaling, and it did so. So with that, we had both a very specific drug and also a drug that addresses a new and hitherto non-addressed biotarget. That was a good starting point for a drug discovery program, an academic in specific. I just want to give you some details how tankerase works. It picks up NAD, which is a redox metabolite that comes from several sources in the cell, mainly glycolysis, but also the OX-phos cycle and mitochondria. And it uses NAD to form a post-translational modification of target proteins. And what is important here, this post-translational modification attracts an E3 ligase, a ubiquitin ligase, which then ubiquitinates target proteins. And this ubiquitination, depending on which kind of linker is used, leads to degradation. So basically, in short words, tankerase regulates protein turnover and protein stability. That's what it does. And with that, it regulates the turnover of the degradosome and destruction complex, because one of the main binding proteins to tankerase is exactly axin, which is the structural protein of the destruction complex. So in a normal situation, when tankerase is active, axin gets parcylated and then through RNF146 ubiquitinated, leading to the destruction of axin, and with that the turnover of the degradosome. Now, if we use an inhibitor and block the catalytic activity of tankerase, then suddenly the destruction complex can't be degraded anymore because it doesn't get ubiquitinated, and we get a huge accumulation of destruction complex, which is so massive that for the first time we can actually see this in light microscopy, and we have explored this further to better understand the structure of this interesting multi-protein complex, and we're just at the beginning of this work, but we do see that we have a filamentous structure that is in close proximity to lipid membranes of the trans-Golgi, and it's a sideline to which I don't want to go in any detail, but we can now use this with photobleaching experiments to study live kinetics of the different components of the destruction complex. And here you see a curve showing, for instance, that beta-catenin very dynamically goes in and out of the complex, while tankerase is very, very stable in this filamentous protein structure. We then went further in what I could call a classical drug discovery program. We identified the pharmacophore by crystallizing the compound with the catalytic domain of tankerase. And that's a work that our collaborator Jens Preben Mort has done, and now Lari Letio is doing. And with that, we have been able to produce a series of analogs that produced a compound that had a highly improved IC50
around 25 nanomolar, which is pretty good. But moreover, also had a glorious per-oral bioavailability, almost 100% in mice. And we were very excited about that the weeks before we did the same experiment in rats and dogs, which showed us that the compound is doing horribly in rats and dogs because there's apparently an active efflux. And so what we have been doing now is we have changed the template to some extent, leaving the central triazole in place. And I am not in a position to show you the new structure, but we have changed the backbone to some extent and get a compound that is amazingly enough even more efficacious and also seems to have resolved the efflux problem. So we're quite optimistic that we will be able, as an academic group, to in fact reach a preclinical stage lead compound. We have to explore this compound, obviously, as the biotarget has been seen as non-addressed in the pharmacological industry, and you can't sell a drug or license a drug to them before they know what the biotarget is worth. So we have been doing a number of things. One is that we have been treating mice with this tankerase inhibitor for several months, and we see that the mice are happy, which is a surprise because Wnt signaling is very important in stem cells. But we do see that the intestinal lining, which is full of Wnt-dependent stem cells, looks perfectly normal. And amazingly, we also see that we have some positive effects on system metabolism with an increased glucose tolerance and improved insulin tolerance. And I don't want to go into any details because this is an ongoing study. But it definitely shows us that we are able to give this compound to animals for a prolonged time. We have been then looking in some detail why the intestinal lining is not affected negatively. And the answer is that although there is a stem cell population at the crypts that is depending on Wnt signaling, and in particular LGR5, as it is named, there seems to be a second stem cell population that is called BMI1 positive cells that can take over, apparently rather perfectly, the functions as long if the main stem cell population is not active, as we show here with lineage tracing experiments in transgenic mice. We then looked into a mouse model that has an APC minus heterogeneous mutation that spontaneously produces adenomas that can develop in colon cancer. And when feeding them with a tankerase inhibitor, we see that we get a great reduction of adenoma formation, and we can do that in more elegant experiments with an inducible APC knockout through tamoxifen, and we see exactly the same. So one of the big issues we had, though, is that when we were running a panel of 660 cells, some cells, cancer cells, some cells were rather responsive and others not at all. And we did not find for several years a fingerprint what identifies sensitive cells and why are they sensitive at all. So what we did then is we did a very broad omics approach and looked for a fingerprint for the sensitive cell lines, and we found BMF coming up as a transcriptional product that's coming up in all the sensitive cell lines, but not in the non-sensitive cell lines. And BMF is a protein that is phosphorylated by a major energy sensor that is called AMP or AMPK, a multiprotein complex that takes up energy signals from different sources. In particular, it measures the ratio AMP to ATP. And what we found, and we were quite excited about, is that all the cells that in our hand were positive, we see a massive phosphorylation of AMPK. So what that means is that we get an energy crisis in the cell upon tankerase inhibition. And why that is so, we just start to understand now. We know that axine is recruited away from the destruction complex by the incoming Wnt signaling. But we also have learned that another protein, in addition to beta-catenin that binds to axine, is precisely AMPK. So what that means is that tankerase does two things in parallel. It coordinates wind signaling by overdriving the incoming wind signaling and stabilizing the destruction complex around the trans-Golgi. But it also coordinates the phosphorylation with that, the activity of the energy sensor AMPK. So what we then did is we looked whether there is any escape mechanism, and as time is running out, there is one, and that is in the TCA cycle. And so we are just learning that if we combine telomerase inhibition, as you see in the graph below here, with a blocker for the TCA cycle, we get some of the sensitive cell lines to entirely stop up growth. While other cell lines that are not sensitive to telomerase, they're also not sensitive to a TCA cycle blocker. We don't yet understand the biology entirely, but we do feel that we have a very interesting predictive biomarker that can tell us why telomerase works in some cases and why not, and that may allow us to, at least to some degree, define patients that may be positively responsive to telomerase inhibition, maybe as a single agent or in combination, for instance, with a TCA cycle blocker. And that this doesn't only work in vitro.
Sorry, 1 minute.
Yeah, I just have 1 minute left. That this also works in vivo, and we have done xenografts, and we see a dose-responsive reduction in growth, accompanied with differentiation, as one would expect if one changes the levels of wind signaling. And this leads me to the conclusion slide. I think we have successfully developed a preclinical lead. We still work a bit on the SAR around that with a novel biotarget that addresses a pathway that hasn't been addressed by the pharmacological industry. Telomerase inhibition shows efficacy in a cancer model by blocking canonical wind signaling and attenuating energy metabolism. And we have defined some parameters, at least, that could identify sensitive cancer cell lines that would respond positively to telomerase inhibition. With that, I just want to show you a last thing. That's our outlook. What we're now doing is that we slowly move into what everybody is excited about, and that's microfluidics, and we try to set up systems derived from iPS cells together with the Stem Cell Center in Oslo, where we can set up models that allow us to identify the different response components in a complex tissue as a cancer would be, but also as the metabolic triangle is that regulates systemic metabolism. With that, I want to thank all my co-workers and collaborators. I want in particular mention Andrei Voronkov. He is sitting here, and he has been working for 3 years in my lab. It was a great pleasure to have him around, and I've been enjoying greatly all the scientific and non-scientific discussions with a very creative mind. Thank you very much.